So this is the second of the narrated PowerPoints on the central nervous system from Chapter 12. And I guess this would be the third section in total of the PowerPoint notes contained within this section. So in this section we're going to be looking at, first of all, the meninges, the outer coverings of the brain and spinal cord. We'll then look at cerebral spinal fluid, review where it's formed from, where it's reabsorbed, and look at its circulation through the subarachnoid space on the outer surface of the brain. We've already looked at the ventricle system on the internal surface. And then the last part of this slide presentation, we'll be looking at the anatomy of the spinal cord, and we'll look in we'll look at major structures, first of all in the gray matter of the spinal cord, and then within the white matter tracts of the spinal cord. So there's 4 structures altogether which provide a degree of protection to the neurons within the central nervous system. First of all, there is the bony protection afforded by the skull for the brain, and the vertebral column in the case of the spinal cord. And then there are the connective tissue coverings, which are, of course, or the membranes which surround the outer surface of the brain and help protect it, which are known as the meninges. And there's 3 different meninges: the dura mater, the outer layer, the arachnoid mater, and the inner membrane being the pia mater. And in addition to the meninges, we have a sort of a water cushion layer, which is formed by the cerebral spinal fluid, which circulates within the subarachnoid space between the arachnoid mater and the pia mater on the outer surface of the brain and the spinal cord. And finally, there's sort of a chemical barrier known as the blood-brain barrier, which helps protect the neurons within the central nervous system, or within the brain at least, from any potential toxins or any drugs or potentially harmful substances that might be circulating in the blood. So the meninges are the connective tissue coverings. They cover both the outer surface or surround the outer surface of the brain and the spinal cord, and they are continuous in terms of there's not one meningeal sort of a group of meningeal structures in the brain and another for the spinal cord. They are in fact continuous. Some of the functions of the meninges, especially within the subarachnoid space, between the arachnoid and the pia mater, they contain important vascular structures which help supply the brain with nutrient. And they also, in the case of the dura mater, they enclose and protect the dural venous sinuses, which are a main part of the venous drainage for the brain. As we mentioned, the subarachnoid space within the meningeal system contains cerebral spinal fluid, which helps, again, provide a protective cushion, also provides some nutrient to the CNS neurons. The dura, the very tough outer fibrous connective tissue layer, in certain areas within the brain, there's normally 2 layers in the dura, and the inner layer of the dura will separate from the periosteum on the surface of the skull, and then forms deep partitions or folds within major fissures in the brain, and helps form really what are in fact partitions between different parts of the brain tissue. And as we mentioned, the meninges in the brain are continuous with the meningeal coverings within the spinal cord. So the 3 meningeal layers: the outer layer, the very tough dura mater, the much more delicate arachnoid mater, this is the middle layer, and then of course between the arachnoid and the pia mater is the space which is filled with cerebral spinal fluid, the subarachnoid space. And finally, the pia mater, which is a very delicate connective tissue layer, which is directly applied to the outer surface of the brain and spine. So the main purpose of this slide is to illustrate the three meningeal layers that are found, in this case, on the surface of the brain. But so we've got the skin of the scalp, and then we've got the periosteum overlying the skull table, or the table of bone that forms the cranial bones here. And then deep to that, we have the two layers of the dura, the layer that is normally applied to the inner surface of the cranial bones, and then the actual meningeal layer, which is the layer which in some cases, in certain locales, will separate and will actually extend down into major fissures to form partitions between. In this case, this looks like it's the superior, this is in the longitudinal fissure, so it's separating the right and left cerebral hemispheres. Another thing to note here, as we mentioned, where there are these deep infoldings of the dura extending down into major fissures, typically within those spaces we find venous sinuses, which are major venous vessels run between the two layers of the dura within these regions, and form part of the major drainage for the major venous drainage of the brain. The subarachnoid space, so deep to the two layers of the dura, we have the arachnoid membrane, a much more delicate connective tissue membrane, and of course below that a space filled with cerebral spinal fluid, the subarachnoid space. And as we mentioned also, there important blood vessels, vascular structures, run through the subarachnoid space as well. And then the final layer of the meningeal layer is the very delicate pia mater, which is directly anchored onto the outer surface of the brain tissue itself. And in this case, again, within the longitudinal fissure, we have the infolding of the dura to form a structure known as the falx cerebri, which is the partition between the two cerebral hemispheres found within the longitudinal fissure. So the dura mater is the outer layer of the meninges. It is by far the strongest and thickest of the meningeal layers. Consists of two layers of dense fibrous connective tissue, or at least in the cranial cavity, that in places these two layers will separate to form, and the infoldings will form the dural venous sinuses within the longitudinal and the transverse fissures of the brain. Again, the dura mater is a very tough, dense fibrous connective tissue. It provides mechanical protection to the brain, and its infoldings help limit movement of the brain and form dividers or partitions within the brain or within the cranial cavity. Three areas where we find these significant infoldings forming partitions, which are created by the infoldings of the dura. One of them is called the falx cerebri, which extends down into the longitudinal fissure, and it effectively divides the two cerebral hemispheres. And then there's a falx cerebelli, much less, a lesser infolding between the two cerebellar hemispheres that runs along the vermis of the midline structure of the cerebellum. And finally, there's an infolding known as the tentorium cerebellum. This is more or less a transverse or horizontal dural fold that extends over the cerebellum within the transverse fissure of the cerebral hemisphere. So this plate illustrates the three dural infoldings, the largest of these being the falx cerebri, the one that extends through the longitudinal fissure, dividing the brain into right and left cerebral hemispheres. And then within these major infoldings, we find large venous vessels which are the major venous drainage from the brain. The superior sagittal sinus and the straight sinus within the dura or within the falx cerebri. But the infolding that extends over the cerebellum within the transverse fissure in the cerebral hemispheres is known as the tentorium cerebelli, literally the tent around the cerebellum. And again, within this fissure, there is also a major venous sinus. The transverse sinus runs through the tentorium. Just another slide to illustrate the, first of all, the dura on the surface of the cerebral hemispheres and within the major sinuses, the infolding of the dura, and then contained between the two layers of dura, in this case, the major venous sinuses, the superior sagittal sinus in the longitudinal fissure, the transverse sinus, which is found within the tentorium cerebelli, the infolding within the transverse sinus. And so again, you can appreciate how thick and tough, what a dense structure this fibrous connective tissue layer surrounding the outer surface of the brain actually is. And it affords obviously a significant degree of protection to the venous vessels which form these venous sinuses. And then in the lower part, down in the brainstem, the dura has been cut away, and what we see is the much more delicate and thinner arachnoid mater, which is covering in this case, the part of the brainstem structures, the pons here and the medulla below. And this is obviously a posterior view, because what we're seeing in here is the cerebellum, which of course is only found in the posterior part of the cranial cavity. So the arachnoid mater, this is the second layer of the meningeal coverings of the brain. It is a much more delicate connective tissue structure. The arachnoid layer is lined by a layer of epithelial cells known as the ependymal cells, or at least the subarachnoid space is lined by ependymal cells. And through the subarachnoid space, we find delicate reticular fibers which extend and which give this layer its name, because it looks like a spider's web, arachnoid being spider-like. It's the arachnoid mater is separated from the dura by a potential space. The subdural space, it's really, in most cases, it's only really a very thin space with just a thin film of fluid in it under normal circumstances. The subarachnoid space, which is found deep to the arachnoid membrane, or the arachnoid mater, is the part of the meningeal layer that contains cerebral spinal fluid, and it also contains larger blood vessels that are going to supply the brain tissue, arteries, and the veins which will drain that circulatory supply to the brain will run along the surface of the brain through the subarachnoid space. And there are specialized structures of the arachnoid membrane known as the arachnoid villi, which in places within the dural venous sinuses will extend through the dura and into the venous sinuses, and these regions are responsible for the reabsorption of cerebral spinal fluid. We're constantly producing it in structures called the choroid plexus, and we reabsorb the cerebral spinal fluid through these specialized projections of the arachnoid membrane into the venous sinuses known as the arachnoid villi. And again, this view that we've seen previously of the various layers of the meningeal layers, but the main thing to draw your attention to here are the arachnoid villi here. where these specialized regions where the arachnoid mater or membrane projects through the dura and into the venous sinuses, mainly in the superior sagittal sinus, which then allows the cerebral spinal fluid to drain off and be returned, the fluid part to be returned back to the bloodstream. So the pia mater is the inner meningeal layer. It's a very delicate and again vascularized connective tissue membrane. Again, the larger blood vessels that run through the arachnoid layer or subarachnoid space will divide up and become smaller blood vessels, which then run within the pia mater and eventually will penetrate down into the surface of the brain in terms of supplying the neurons in the brain. And again, the other thing to mention or emphasize with the pia mater, this connective tissue layer is directly applied and densely sort of held, if you like, or melded to the outer surface of the brain and the spinal cord. So the cerebral spinal fluid, or CSF, this is a filtrate of the blood. Specialized ependymal cells filter the capillary blood and then form, allowing the fluid part to pass through, forming the cerebral spinal fluid that is released into the ventricles and eventually will circulate through the subarachnoid space. Composition of CSF, it's basically water, but it shouldn't contain any blood cells and shouldn't contain any platelets, so no formed elements of the blood. And then the protein content is considerably lower than the protein content in the blood. And there are some differences in terms of ion concentrations as well, especially the potassium ion is found in considerably lower concentration in CSF than would be found in the extracellular fluid and within the blood. It does contain nutrients, however, free fatty acids and glucose, which can serve as nutrients for brain tissue, are capable of circulating through and are part of the cerebral spinal fluid. And so CSF, part of its function is that it does provide some nutrition to neural tissue. So again, the purposes of the CSF, it gives it allows the brain and spinal cord to basically float within a fluid environment. That fluid layer or water layer provides a cushion protecting the CNS from blows and other forms of trauma which may affect the skeletal tissues, which help surround the central nervous system. And finally, the CSF, as we mentioned, does provide some nourishment to the brain tissue, and it is also capable of carrying some of the chemical signaling molecules that may affect or modify neural function. So this plate sort of provides a simplified version of CSF, where it's produced and finally where it's drained off. So if we start off at number 1, the choroid plexus specialized ependymal cells are responsible for filtering the blood and forming the CSF, which is then released into the ventricles of the brain. All 4 ventricles in the brain produce CSF, the 2 lateral ventricles, the 3rd ventricle here in the diencephalon, and then the 4th ventricle down in the lower part of the brain between the brainstem and the cerebellum behind. So all 4 chambers have choroid plexus that produce cerebral spinal fluid by filtering the blood. The CSF then circulates through the ventricular chambers from the lateral ventricles to the 3rd ventricle through the cerebral aqueduct down into the 4th ventricle. From the 4th ventricle, there is a midline aperture and 2 lateral apertures by which the cerebral spinal fluid can escape the ventricular system and be released into the subarachnoid space. And so the CSF then circulates through the subarachnoid space surrounding the spinal cord and surrounding the outer surface of the brain. And in certain regions within the brain tissue, the arachnoid membrane projects through the dura and into the venous sinuses that run through the dura in the dural folds, particularly in the superior sagittal sinus, in the longitudinal fissure, and in these regions the CSF is drained off and returned back to the blood. And these specialized regions again were responsible for the draining of CSF, or the return of CSF from the subarachnoid space back to the bloodstream. These regions are known as the arachnoid villi. So the choroid plexus, this is the region, specialized types of ependymal cells which are responsible for filtering the blood and producing cerebrospinal fluid. There are choroid plexi found within the upper part of each of the ventricles within the brain. And they're basically, again, they filter capillaries, and they're specialized ependymal cells. There's normally about 150 mLs produced every 8 hours, so roughly 500 mLs per day are produced in total within the 4 choroid plexi in the ventricles. And this is just an illustration. So in this case, the ventricular cavity would be around the periphery here. This is showing you the capillary blood vessel and how the ependymal cells sort of serve as a filter, allowing the water portion of the blood, all right, to filter through and to form CSF. And again, it's mostly water, but it does contain glucose, oxygen, and some of the ions which would normally be present within the blood. Okay. The cellular elements and waste products aren't allowed to be filtered through, so they're retained within the bloodstream. And finally, the final sort of protective layer, helping protect the CNS neurons, the blood-brain barrier. Again, this is a filtering mechanism which helps prevent substances that would be harmful to neurons within the bloodstream from actually reaching the interstitial fluid and the neurons of the brain. It's designed to sort of separate the neurons, or form a filter, if you like, between the neurons and the capillary blood vessels that are supplying nutrient to the brain. So there are 3 elements to the formation that make up the blood-brain barrier. One of them is the continuous endothelium of the capillary walls. So these are abnormally tight junctions between the endothelial cells in the capillary walls of the blood vessels supplying the brain. And again, that restricts leakage of substances or the movement of many substances between the blood and the interstitial fluid of the brain. There's a basal lamina, which is again relatively thicker than in many tissues in many areas of the body surrounding these capillaries, and again that helps prevent diffusion of substances between the blood and the interstitial fluid of the brain. And finally, there are the astrocytes. The extensions of the astrocytes, or so-called feet of the astrocytes, which wrap themselves around capillary blood vessels and provide a further barrier in terms of allowing substance to move freely between the blood and the neurons. And this slide is just showing you the astrocytes and how their cellular extensions sort of wrap themselves around the capillary blood vessels and form part of this separating layer between the blood vessels and the neurons that it's supplying nutrient to. So this is a selective barrier. It allows nutrients to move via facilitated diffusion between the capillary blood vessel and the neurons. Metabolic waste products, most proteins, potential toxic elements like ammonia and urea, many drugs, some of the amino acids, and potassium actually is largely denied, and so it's restricted from moving out of the blood and to the neuron. Most fat-soluble substances are capable of moving freely between the blood vessel and the neuron, and this would include alcohol, nicotine, and a variety of anesthetics, or drugs that we use as anesthetics to modify or depress neural functioning. In places the blood-brain barrier is absent, there are areas where we need to monitor conditions in the blood. It's important for the brain to know the composition and, for example, the presence of certain products in the bloodstream. And so in these regions where we have a need for this function, and there is an absence of the blood-brain barrier, the hypothalamus is a good example of a region which largely lacks a blood-brain barrier. The glandular tissues in the central nervous system, the pituitary and pineal gland, are other areas which have a necessity to monitor makeup of the blood and the presence of certain chemical molecules within the blood, and therefore they lack a blood-brain barrier. So the spinal cord, we're going to look at its basic anatomy and the protection in various locations that it is afforded. In terms of location, the spinal cord begins at its upper end at the foramen magnum. Distally, the spinal cord ends at usually at the disc space between the L1 and L2 vertebral bodies. That's how far it extends distally in the adult. So again, it does not extend completely through the spinal or vertebral canal. It ends sort of almost at the thoracic lumbar junction, just extending barely into the lumbar spine. The functions of the spinal cord, it's there to provide two-way communication between the lower motor neurons or lower neurons, both sensory and motor, and the higher brain centers. It contains gray matter, which is responsible for generating spinal reflex responses. So in terms of protection, it's largely the same as the brain. Again, we've got bone tissue, in this case the vertebral column. The meningeal layers, of course, are continuous with the brain. So the three layers of the meninges, and again, circulating in the subarachnoid space, we've got the cerebral spinal fluid there to help provide protection. The blood-brain barrier is not nearly as well developed within the spinal cord as it is within the brain. So that is a less effective, much less effective protection within the spinal cord than it is in the brain. The epidural space. So this is the physical space outside the three meningeal coverings, but within inside the bony confines of the vertebral column. And the epidural space contains typically a layer of adipose cells and blood vessels found within this space, and again that provides a cushioning effect and a further layer of protection in terms of helping protect the spinal cord within the confines of the vertebral column. The CSF, of course, circulates within the subarachnoid space of the vertebral column or around the spinal cord, and at the inferior end, we mentioned how the spinal cord typically ends at the level of L1 and L2, but the dura, the two outer membrane layers, the dura and arachnoid membranes extend down to the sacrum well beyond the terminus of the spinal cord at L1 and L2. And so this is a region where we can safely do a lumbar puncture or spinal tap, or we can insert a needle into the subarachnoid space, withdraw cerebral spinal fluid in order to, typically as a form of lab testing, used, for example, to detect presence of infection in some cases within the meningeal spaces. The spinal cord, where it comes to an end in the upper lumbar spine, it sort of gradually comes to a tapering point, and this part of the spinal cord, which represents the really the sacral part of the spinal cord, where it tapers to this sort of fine tapering endpoint, is known as the conus medullaris. And at its very distal end, there's a fibrous membrane that extends from the distal end of the spinal cord called the filum terminale. And this fibrous membrane or connective tissue layer extends right down to the surface of the coccyx at the lower end of the vertebral column. And the filum terminale is covered continues to be covered by the pia mater that it also extends down to the coccyx. And so this serves as an anchoring point for the distal end of the spinal cord. In addition to that, there are, throughout the length of the spinal cord, there are extensions where the pia mater extends from the actual outer surface of the CNS tissue and runs outwards to the inner layer of to attach to the dura, and that helps again stabilize or anchor the spinal cord securely within the meningeal layers. So this view is a midline sagittal section, all right, from the lumbar spine. It's showing the lower lumbar vertebrae, L4, L5, S1, and shows the typical location for a spinal tap or a lumbar puncture, which is to evaluate the composition of the cerebral spinal fluid. And again, in this case, there's a lot of extra room within the lumbar spinal canal because the spinal cord has ended above this. The only neural structures that continue through this space are the nerve roots, the ventral and dorsal roots that will form the spinal nerves for the lumbar and sacral spinal nerves. And so again, and also through here we've got the filum terminale, which is this sort of green colored structure extending down, and eventually that extends right down to the surface of the coccyx. And the outer two coverings of the spinal cord, the dura and the arachnoid membrane, extend down to the level of the second sacral vertebrae. In this case, this is I guess a longitudinal section of the spinal cord where the actual the outer superficial layers, the dura and the arachnoid membranes, have been sort of cut away and folded back to show you the actual structure of the cord. The cord in a couple of areas is thickened or enlarged. There's a cervical enlargement where the nerve structures that will supply the upper limb come off the cord, and there's a lumbar enlargement where the extra spinal tissue related to supplying the lower limb comes off the cord. So in those regions the cord becomes thicker or larger due to the increased presence of neurons within the spinal cord in those areas. And of course the spinal cord ends distally at the level of L1, L2, typically in what is known as forming what is known as the conus medullaris as it comes to a tapering point. Below that, the nerve roots that will form the lumbar and sacral spinal nerves continue through the lumbar spinal canal and into the sacral spinal canal. And because of these are made up of sort of a number of different fiber groupings, it got the name cauda equina because it looks like the very dense coarse hairs in a horse's tail. And so it's the cauda equina, which is made up of the lumbar and sacral spinal nerves, which continues beyond the spinal cord through the lumbar and sacral spinal canals. The next few plates are just detailed photographs of different regions within the spinal cord to give you an idea of its actual physical structure. So again, you can see here the cervical spinal cord, and this would be the cervical enlargement, where some of the nerve roots that will supply the upper limb are coming off the cervical cord. This is from a posterior point of view, so this is the cerebellum shown in here. This is part of the cranial dura mater covering the cerebellum, the infolding between the two cerebellar hemispheres in here of the dura. The lower, this would be part of the brainstem in here, the pons, the medulla in here, and the first of the cervical spinal nerves forming in here. These are, and again, these come off as rootlets. Several of these, this would be from the dorsal side, and the dorsal side is the sensory side. So these would be the sensory roots helping form the spinal nerves at the cervical levels shown coming off here. Each of these would represent one, the sensory root at one vertebral level. And the other note here, again, the pedicle, this is the part of the vertebrae that extends posteriorly around the spinal cord. So the pedicle, the point at which the vertebral arch extends posteriorly to surround the spinal cord, the vertebrae has been cut at that point. So this is showing you part of the thoracic spinal cord, or a section of thoracic spinal cord. Again, the vertebral arch has been cut. This would normally completely enclose the spinal cord from a posterior perspective, but the bones have been cut to reveal the outer coverings of the dura, the outer coverings of the cord. The dura mater and arachnoid mater have been cut away. Again, you can see the same basic structure. This is, again, a posterior view, so these are what we're seeing here are the rootlets, which will form the dorsal root, or which is the sensory root of a spinal nerve. Now, on this side, at a deeper level, though, you can also see the ventral root here coming off the anterior side of the spinal cord down in through this region. And so these two together, the dorsal root, which is sensory, the ventral root, which is the motor fibers coming off the ventral or anterior part of the cord, will come together and will form a spinal nerve down in this region in here, which will then exit the vertebral column through the intervertebral foramina. And again, the coverings of the cord, on the anterior surface here, the dura mater, the tough outer covering. And the superficial, at a more or at a higher level, the separation of the arachnoid mater in here. And the denticulate ligaments, again, we mentioned these. These are areas where the pia mater extends out from the surface of in sort of a ridge or sawtooth-like arrangements all the way up the spinal cord, running from the outer surface of the spinal cord and anchoring to the inner surface of the dura. So it again sort of holds the spinal cord in place within its meningeal coverings. So this is showing us the lower end of the spinal cord as it's drawing, or as it's reaching its inferior extent. So this is again at the level of L1 and L2 down here where the spinal cord is terminating. So this is the cord in here. You can see the sort of whiter colored region in here as it comes to a finely tapered point. This is known as the conus, all right, and represents the sacral levels of the cord. And then below that, it's just a fibrous extension attached to the distal end of the spinal cord, which is known as the filum terminale, which extends right down to the coccyx at the inferior end of the spinal cord. And you can also see here the formation of the cauda equina with the rootlets, or the roots, which will form the spinal nerves coming off respectively the anterior and posterior surface of the cord and continuing to run down through the lumbar spinal canal, where they will join together and form spinal nerves at each level. And of course the structure that these roots of the lumbar and sacral spinal nerves form, because again they look like these thick strands of hair, they were called the cauda equina. The spinal cord itself is divided up into 31 different spinal segments. The spinal nerves which come off of the spinal cord are part of the peripheral nervous system. Again, once they've exited the cord, they are considered as outside the central nervous system. So there's 31 pairs of spinal nerves that come off the spinal cord, and they're in a couple of regions, as we mentioned. There are enlargements or thickened areas of the spinal cord where the extra neurons related to supplying the upper limb in the case of the cervical enlargement and lower limb in the case of the lumbosacral region provide a thickened area of the cord. The cauda equina, again, we mentioned it with the last slide. This is a collection of the nerve roots that will form the lumbar and sacral spinal nerves. They continue to run inferiorly below the level where the spinal cord is ended through the lumbar and the sacral spinal canals. We're now going to look at the cross-sectional anatomy of the spinal cord. There's two longitudinal grooves that partially divide the cord into right and left halves. There's a ventral anterior median fissure, and then there's a less deep indentation on the posterior side called the median sulcus. And there's an area of the gray matter of the spinal cord is concentrated in the central region, and it forms sort of a butterfly configuration, and connecting the two sides is what is called the gray commissural fiber, which consists of cell bodies and unmyelinated neurons, which connect the right and left sides of the cord. So this is a cross-sectional view showing the spinal cord sort of in situ, sitting in this would be actually a cervical vertebrae, because it's got the transverse foramina in it here. But it's showing the spinal cord sort of in situ as it would sit within the vertebral canal or spinal canal. So again, within this region, we've got the epidural space, this layer of fat and vascular structures which surrounds the meninges and the spinal cord within the vertebral canal. And then the three meningeal layers, the outer layer being the dura, middle layer being arachnoid, and innermost layer directly applied onto the surface of the spinal cord, the pia mater. And then the thin subdural potential space and the wider subarachnoid space through which cerebral spinal fluid circulates. And then the other thing we can see in here is just, which is blown up more significantly in the next slides, but these median fissures on the anterior side and a median somewhat less identifiable indentation in the posterior midline called the median sulcus. Okay, the butterfly configuration, which represents the gray matter of the spinal cord, gray matter being the concentration of cell bodies and unmyelinated axons. So this is kind of the, if you like, the thinking side or the decision-making area as such within the cord, the very small central canal in the midline, and then the thin layer of gray matter which connects the right and left sides of the cord, called the gray commissural fibers. And then the rest of the spinal cord, this whole area which is represented as white, and it actually is white, this is referred to as the white matter of the cord and consists of the myelinated fiber tracts, most of which are running in a vertical direction, connecting the neurons of the spinal cord with neurons in the brain. And the two parts that will form the spinal nerve coming off of the anterior part of the cord is a series of rootlets which form what is called the ventral root of the spinal nerve, which is the motor root of the spinal nerve, and coming off this dorsal extension of the gray matter is the dorsal root, which are the axons of the primary sensory neurons, which are going to come in and synapse within this region, this dorsal part of the spinal cord. So dorsal root and ventral root, which join together to form the spinal nerve. So again, a cross-section view of the spinal cord with its meningeal covering, so dura mater, arachnoid, and of course pia mater down here. And illustrating again how the spinal nerve is formed. There are fibers which come out of this anterior part of the gray matter known as the ventral horn, and the rootlets or axons which come out of this collect to form a common neural structure known as the ventral root of the spinal nerve, and this is the motor side. And then the same thing basically happens on the posterior side, except these are incoming fibers coming from sensory receptors out in the periphery and conducting information into the central nervous system or spinal cord. They all collect and come into the posterior part of the gray matter, which is known as the dorsal horn of the spinal cord. And these are sensory, the primary sensory neurons, the axons of which will come into the dorsal horn, and most of them will synapse there. Okay, and there is oftentimes also a sort of lateral expansion of the gray matter or extension of the gray matter known as the lateral horn, and these are autonomic motor fibers, particularly in the thoracic and upper lumbar regions that we find this. Okay, so in the gray matter, the posterior extension is the dorsal horn. The anterior enlargement of the gray matter is known as the ventral horn. Dorsal horn is always sensory neurons and interneurons. Ventral horn is always motor neurons. The lateral horn, where it is found, and it's found in the thoracic and upper lumbar regions, is the region where we find autonomic motor fibers. So that's the gray matter. It's got this butterfly type of configuration, and it forms the central part of the spinal cord. It is surrounded by white fiber tracts or white matter fiber tracts, which are the vertically running tracts, allowing for communication between the motor neurons or the neurons within the spinal cord and higher brain centers. The white matter is divided up into three main columns. Those three main columns are known as funiculi. There's a dorsal funiculus, which is basically represented from the dorsal midline out to the area where the dorsal root of the spinal nerve comes off. There is a lateral funiculus, which is the area between where the dorsal root comes off and where the ventral root comes off the ventral horn of the cord. So that's the lateral column or lateral funiculus in here. And then there's a ventral or anterior funiculus, which is the area between where the ventral root comes off the ventral horn and the anterior midline in here. So this region in here would be the ventral or anterior funiculus. And then for the dorsal root ganglion, this is the area where we find the cell bodies for the primary sensory neurons. Okay, again, a ganglia should mean a collection of cell bodies found outside the central nervous system. So because these aren't contained within the cord itself, these are considered to be outside the central nervous system, even though they're within the vertebral column. Because the vertebral column would basically end at the outer extent of where the dura ends in here. So the nerve would be exiting through the intervertebral foramina right in this region in here. Okay. But this dorsal root ganglia represents the cell bodies for the primary sensory neurons, and then their axon continues into to forming the dorsal root of the spinal nerve, which will then come in and synapse within the dorsal horn of the spinal cord. Okay, we've mentioned these structures in the previous diagrams, but the dorsal horns, these represent, these largely contain interneurons that will receive both the somatic and visceral input carried by primary sensory neurons. So the primary sensory neurons coming either from somatic or visceral structures will come into the dorsal horn and will then synapse with interneurons or integrative neurons within the dorsal horn of the spinal cord. The ventral horn of the spinal cord contains some interneurons and also contains the somatic motor neurons. Somatic motor neurons are the ones that are going to supply skeletal muscle tissue. And the motor axons from the somatic motor neurons will exit the cord, forming the ventral root of the spinal nerve. And then, of course, the lateral horn, which is found in the thoracic and upper lumbar regions, this will contain sympathetic neurons, part of the autonomic nervous system. The dorsal roots, the dorsal root contains the sensory axons of the primary sensory neurons, i.e. the ones that are coming from receptors outside the central nervous system, carrying the signals into the spinal cord. And the dorsal root of the or what we call the dorsal root or spinal ganglia are the cell bodies of the primary sensory neurons, which again are considered as being outside the CNS. So the text of this slide is referring to the diagram in the next slide, which I'll jump to in just a moment, but again, this is just trying to show you as a representative sort of color-coded arrangement what areas contain somatic sensory or receive somatic sensory input, what areas receive somatic or visceral sensory input coming from the internal structures of the body, the areas where we find visceral autonomic fibers, which would be that lateral horn region, which are going to supply smooth muscle, cardiac muscle, and glandular tissue, and finally the areas of the cord which are the ventral horns of the cord, which will contain the somatic motor neurons, or what we commonly call lower motor neurons, which are distributed to the skeletal voluntary muscle. And so if we jump ahead, again, those structures, again, so the more peripheral parts of the dorsal horn, this posterior extension of the gray matter, is where the somatic sensory fibers, primary sensory neurons, will terminate, and then a little farther centrally within the dorsal horn is where the visceral somatic sensory fibers will terminate. And then in the lateral horn, this is where we find the motor neurons of the autonomic system, the sympathetic system in particular, visceral motor fibers, and then the somatic motor fibers, which represent the lower motor neurons, which are going to supply skeletal muscle tissue, occupy the main part of the ventral horn of the gray matter in the cord. And so the motor fibers exit via rootlets, which will form the ventral root of the spinal nerve, the motor root, and the incoming sensory fibers will form the dorsal root of the spinal cord, and of course the cell bodies for those sensory neurons are located in the expansion of the dorsal root just inside the vertebral column, but outside the spinal cord, which is known as the dorsal root ganglia. So we're now going to look at the white matter making up the spinal cord. These are mainly myelinated, or the predominantly myelinated fibers, because many of these fibers tend to run long distances. And again, the myelinated fibers are afforded a degree of protection and a faster rate of conduction, which improves their efficiency. So there's 3 fiber groupings, or we can divide these up into 3 functional groups. The ascending fibers, which are mainly sensory fibers, which are running from spinal cord levels up to higher brain levels, be that in the brainstem or the thalamus or onto the cerebral cortex. And then descending fibers, these are primarily efferent fibers that are running from the brain down to the cord, or between from higher to lower levels in the cord, and these are primarily motor fibers, which are carrying signals to the lower motor neurons in the ventral horn, or autonomic fibers that are going to then signal muscle tissue or glandular tissue. And finally, the transverse fibers, these are the ones that tend to run through the commissural area that connect or communicate between the 2 sides of the spinal cord. So as we mentioned, the white matter is divided up into 3 main columns or funiculi on each side. There's a dorsal column or posterior column. There's a lateral column, which is the area between where the dorsal root comes off and the ventral root comes off anteriorly. And then there's a ventral funiculus, which is the area between where the ventral root comes off and the anterior midline. And then within each of these areas, there will be various tracts or spinal tracts which run, and again, in each of these tracts, these will be composed of axons which have similar functions and which have similar sort of start points and end points typically. And if we jump ahead a couple here, so this would be a good illustration of those tracts where we've got the posterior funiculus in this area, or the dorsal columns. These are sensory, and again, anything in blue is sensory, anything in red is motor within these columns. So the posterior funiculus is always sensory, purely sensory. The lateral funiculus, again, is the area between where the dorsal root fibers come off, or the dorsal root of the spinal nerve comes off the cord, and the area where the ventral root comes off. So it's the region from here to here. This is the lateral column, and it is a mix. It's pretty much an equal mix of both sensory and motor fibers within this area. And then the anterior column or ventral column is the area between where the ventral root comes off and the anterior midline, and it is predominantly motor, as you would expect, because the ventral horn here is the motor side of the in terms of the gray matter. But there is one important sensory tract, the ventral spinal thalamic tract, that does run through the ventral or anterior funiculus. So those are the 3 funiculi, and again, within each of these funiculi, we will have various tracts, which are individual groups of axons which tend to have similar functions and have similar origin points and final destination points. So another point that we need to make here is just, we're not going to ask you guys to remember names to any great degree in here, but in terms of, in many cases, the naming of these tracks will tell you a great deal about, where the tract is located and where its start point and end points are, which you can often then decipher, for example, whether it's a motor tract or a sensory tract. So the naming of these tracts, there's often three different parts, or two different words to the name. For example, lateral corticospinal tract. So the first word, the lateral portion here, or it might be ventral or it might be dorsal, but it tells you what funiculus or what column the tract is running in. So is it in the dorsal column, is it in the lateral column, which this one is, or is it in the ventral column of white matter? Corticospinal, the second word in the name, again, typically will tell you the origin point for these fibers and their finish point or destination point. So corticospinal, the origin part is the first part, the destination is the second part. So corticospinal in this case is telling you that this is coming from cortex, meaning the cortical part of the brain, the cerebral cortex, and it's ending at spinal cord levels. So it's running from the cortex down to the spinal cord. So this is a descending tract, clearly. And again, that would mean it's a motor or efferent tract. Okay. So lateral corticospinal tract is a motor tract which runs through the lateral funiculus, okay, starting from the cortex and ending at spinal cord levels. Some general rules in terms of these white matter tracts or pathways. So most of these major spinal tracts are part of a multi-neuron pathway. Typically, there will be several neurons involved in transmitting a signal going from the motor cortex to ultimately end at spinal cord levels. Decussation is a terminology that is used. Decussation means crossover from one side to the other. And almost all of these tracks, at some point or other, the information will cross because, as we've mentioned, cerebral cortex, which is the either the origin point or destination point for much of this information, but virtually all of the input or output from the cerebral cortex ends up crossing at some point, affecting the opposite side of the body. Relay is just it's an area of synapse connection, and it often consists of the signal being passed through several different integrative or connector neurons at this relay. And then somatotopy, this is a characteristic whereby the fiber groupings within the axons within these tracks will maintain a very precise spatial relationship. So axons coming from one particular region will all tend to run on one side of the tract, and then they end on, at their destination they will signal integrative neurons, and so that signal is not only transmitting information but is also carrying information about the spatial relationship where those axons originated from. And symmetry, again, these pathways, both the sensory and motor pathways, are basically paired structures, and the structure of the white matter of the cord, like the structure of the brain in general, is symmetrical. So we just looked at this, describing the funiculi, the posterior funiculus, the lateral funiculus in here, and the anterior funiculus in this region. Posterior funiculus is purely sensory. Lateral funiculus is an equal mix of sensory and motor tracts. The sensory in this case again being blue, the motor being red, and the motor, or the anterior funiculus, is mainly motor, but there is one important sensory tract, the ventral spinal thalamic, in here. And again, the gray matter and white matter, or the gray matter in this area, again, the dorsal horn of the cord is the sensory region of the cord. The ventral horn of the cord is where we find somatic motor fibers. And these primary sensory neurons come into the dorsal horn, and the dorsal root comes into the dorsal horn of the cord. And in terms of the output end, the lower motor neurons in the ventral horn, their axons will form the ventral root of the spinal nerve, which is the motor root of the spinal nerve. And again, we haven't discussed individual tracts at all. We will a little bit later on, but again, all of the red tracts in here are descending or motor tracts carrying signals from the brain, eventually that will be distributed to the lower motor neurons in the ventral horn of the spinal cord. And these blue colored tracts are sensory tracts, which are carrying ascending signals. They're ascending to brain levels, and eventually this information will be transmitted to sensory areas within the cerebral cortex, or in the case of spinocerebellar tracts, the cerebellum. Okay, we're now going to look at ascending pathways, so sensory information. Most of these ascending pathways involve at least 3 different neurons going from the primary sensory neuron, which carries the initial signal coming from the receptor, to get up to the cerebral cortex. And there may well be more than that, but there's a minimum of 3 neurons in transmitting that signal. So the first order neurons are identified as primary sensory neurons. These are the only ones which are truly sensory neurons, okay, by our definition. And so all of the others, the second order and third order neurons which are carrying sensory signals, are actually interneurons or integrative neurons. So these first order or primary sensory neurons conduct the impulse from receptors located on the body surface, or from visceral tissues of the body, or from proprioceptors. They come in with the spinal nerve. They enter via the dorsal root of the spinal nerve, and they will eventually enter the spinal cord, where they will then synapse with usually in the dorsal horn of the spinal cord, with secondary sensory neurons, or second order neurons. Again, the majority of the sensory neurons make their synapse or initial contact point in the dorsal horn of the spinal cord, which is the sensory region of the spinal cord, or deals with sensory information. The neurons that the primary neurons will synapse with are actually integrative neurons. Technically, they are not sensory neurons. Okay. And when we say they make a synapse, we're using that in a very general sense. This is actually a relay. So that signal will get transmitted through several different integrative neurons within the dorsal horn before it excites what we call the second order sensory neuron, which is then going to enter an ascending tract and carry the signal upward to the brain. Okay. The dorsal column sensory neurons are a bit of an aberration. While they do make some synaptic connections in the dorsal horn, the fibers do not end there. the main part of their fiber grouping continues and enters the dorsal column ascending tracts, and they ascend all the way up to the level of the medulla before they end in sensory nuclei within the medulla. So these are very long axons, or very long fibers. All right, some of these are coming, for example, from the foot and run all the way from the foot or the tip of the big toe at the greatest extent all the way up through the leg, okay, enter the spinal cord at either lumbar or sacral levels, all right, and then instead of ending at that level in the dorsal horn, they enter one of these ascending dorsal column tracts, and they ascend all the way up to the base of the brain, in the lowest part of the brainstem, the medulla, before they end. So these could easily be, 4 or 5 feet, and if you're very tall, 6 feet long. So the second order neuron, and we refer to these as secondary or just second degree, if you like, sensory neurons. And again, this is confusing. We call them sensory because they're transmitting sensory information, but technically these are integrative or interneurons. And so their cell body is located within the dorsal horn of the spinal cord typically, or in the case of the dorsal columns, they're going to be in the medullary nuclei. If this is a sensory signal being transmitted through the dorsal column pathway. So, but most of these second order neurons, sensory neurons, enter an ascending tract and will carry their information upwards through the spinal cord and will extend up to the thalamus. Some of the secondary proprioceptive axons, those that are in, for example, spinocerebellar tracts, do not ascend to the level of the diencephalon, but they instead will be relayed directly into the cerebellum. Much of the proprioceptive input will go directly either to vestibular nuclei or to the cerebellum instead of to the thalamus. And there are a large number of these secondary sensory axons which terminate in reticular formation nuclei in the brainstem. And again, it's worthwhile emphasizing that at every one of these relay points or synapse points within the ascending pathway, much of the sensory signal ends up getting filtered out, okay? Meaning that it's not strong enough or not considered important enough to generate an action potential in the next neuron in the chain, and therefore at this point the signal will simply die out, okay? A good example of this is where, you're sitting there reading this or listening to this, and you've got you're wearing clothing, you might be wearing shoes, but and with that there are sensory receptors which are being stimulated by the presence of the clothing on that you're wearing on your body, or the shoes you're wearing on your foot. But at the same time, we're not, you're at conscious levels in your brain, you're not aware of that information, and you're certainly not responding to that information. The only time we're actually aware of it is when we initially put the clothes or the footwear on, and then what happens is basically the cortex initially registers the fact that, all right, I've got this material on my body which is sending, activating touch receptors, and that information is being relayed up to the brain. But once that initial signal is sent, then subsequent signals are basically filtered out before they reach levels of conscious thought. All right, and that's true of, again, the great majority of sensory information that's being generated from various receptors in the body. Most of it never reaches levels of conscious thought. It ends up being filtered out at some part of this ascending pathway relay system. Okay, and then there's third order sensory neurons, or tertiary sensory neurons. Again, these are integrative or interneurons. Many of these originate within the nuclei within the thalamus, which is a major relay point going, eventually relaying signals up to the cerebral cortex. So the cell bodies for these neurons are usually located in thalamic nuclei, and then the axon emerges from these cell bodies and enters another fiber tract, ascending tract, running from the thalamus up to the somatosensory cortex. If the information is being relayed into the cerebellum, then that information isn't relayed onwards from there. Okay, and the great majority of the proprioceptive sensory input is subconscious. We never are aware of it. There's some proprioceptive signals that are relayed through the dorsal columns, which does reach levels of conscious thought, but much of it we are never consciously aware of. It's dealt with within the cerebellum, and we don't have a conscious awareness of what's going on in the cerebellum. In the case of the second order neurons, which terminate in the brainstem reticular nuclei, in some cases there will be third order axons that will arise from the brainstem reticular nuclei and will ascend to the thalamic sensory nuclei, and then some of these neurons will then be sent onwards to the somatosensory cortex. So in this case there would be a fourth order sensory neuron which would be involved with transmitting that information onwards to the somatosensory cortex. So we're going to briefly talk about three main ascending tracts or ascending pathways which carry sensory information through the spinal cord and up to higher brain centers. Two of these sensory pathways will transmit information to the sensory cortex via the thalamus. Those two are the dorsal column medial lemniscus pathway. This pathway provides or carries information concerning fine touch, and fine in this case means well localized touch and pressure sense, vibration sense, and some conscious proprioceptive information. That's what this tract carries, and it will relay that information through the thalamus and onwards to the primary somatosensory cortex. The spinal thalamic pathway, this pathway will transmit pain and temperature sense in a tract called the lateral spinal thalamic tract, and then it's got a second division which transmits through a more anteriorly placed sensory pathway, the ventral spinal thalamic tract, and this pathway transmits crude touch and pressure sense, which is poorly localized touch and pressure sense, tickle and itch sensation, and sexual sensation carried in this ventral spinal thalamic tract. And again, it will relay its second order neurons go to the thalamus, and third order neurons will be relayed onwards to the somatosensory cortex, carrying pain and temperature sense in the lateral spinal thalamic, crude touch and pressure, tickle and itch, and sexual sensation in the ventral spinal thalamic. And finally, the third fiber tract is a little bit different. It's the spinal cerebellar tracts. These do not ascend to the somatosensory cortex. Okay, these tracts terminate in the cerebellum, and they carry subconscious proprioceptive information up to the cerebellum, which will be utilized then in helping coordinate and direct motor function. So this is just a diagram representation, and these aren't the best diagrams. It's a lot of information to try and represent here on one plate, but on the right hand side here we've got the dorsal column medial lemniscus pathway, which is carrying in this case touch sensation or proprioceptive sensation. These fiber tracks do, again, this is the one that doesn't synapse at the level of entry in the spinal cord, but its main fiber continues onwards and enters the ascending or dorsal column pathway, which ascends all the way to the level of the medulla before making its first synapse with the two nuclei for the dorsal columns, which are located, sensory nuclei, in the medulla. And then the second order neurons will emerge from the medulla and cross over the opposite side, ascend through a tract called the medial lemniscus to the thalamus. And then the third order neuron will be relayed from the thalamus up to the somatosensory cortex, okay, or primary cortex. And on the left-hand side here, in the lighter colored aquamarine color, these are proprioceptive inputs, in this case coming from the muscle spindle, but it could be Golgi tendon organ, it could be a joint receptor or pressure receptor. This information comes into the dorsal horn of the spinal cord, will make an initial synapse in the dorsal horn of the cord. Second order neuron will emerge out of the dorsal horn and transmit that, carry that information upwards in a spinocerebellar tract, and that will eventually terminate in the cerebellum, where that will be utilized in helping regulate motor activities, or in some cases helping us maintain balance during movement. Okay. And the spinal thalamic pathway, so again, this is ascending, because the first part of the name is spino and ends at the thalamus and the diencephalon, so it's an ascending or sensory tract. There's two divisions to this, the lateral and ventral spinal thalamic tract. The lateral spinal thalamic tract transmits pain and temperature sense. The ventral spinal thalamic transmits poorly localized touch and pressure sensation, and also tickle and itch and sexual sensation. Okay, and these tracts ascend, make an initial synapse in the dorsal horn of the spinal cord. Second order neurons will cross to the opposite side and enter one of these ascending tracts, either the lateral spinal thalamic or ventral spinal thalamic. They will ascend to the level of the thalamus and then make another relay in the thalamus, and third order neurons will then transmit that information on to the primary somatosensory cortex. And this is just a diagram representation for the spinal thalamic tracts, in this case temperature sensors or pain receptors. So this is the lateral spinal thalamic tract we're detailing here, carrying pain and temperature sense. These neurons come in, they make an initial synapse in the dorsal horn at the level where they enter the spinal cord. The second order neuron, it goes through several integrative neurons, but the second order neuron will then emerge from the dorsal horn, cross the opposite side, enter the lateral spinal thalamic tract, and will ascend to the level of the thalamus. And then in the thalamus it will make another relay, and third order neurons will then emerge from the thalamus to carry the information up to the somatosensory cortex. I'm going to add in a fourth order or a fourth sensory pathway here, the trigeminothalamic pathway, which is significant for you guys working in the facial and oral area. Again, this tract is only found in the brainstem, so it's not found in the spinal cord the way the other three tracts were. But this is the sensory pathway which carries sensation from the facial area and from the oral cavity, and this is the way it arrives up to levels of conscious thought. So the primary sensory neuron in this case is carried in branches of the trigeminal nerve. Some of these fibers will then synapse, so they carried in the trigeminal nerve and comes in to the primary these fibers will initially, most of them will synapse with secondary sensory neurons in the pons of the brainstem. Others descend actually to the medulla, where they will synapse with second order sensory neurons. The second secondary sensory neurons, in this case, cross to the opposite side of the brainstem and ascend to the thalamus in a separate tract, ascending tract known as the trigeminothalamic tract. Okay, particularly pain and temperature sense utilizes this tract. Touch and pressure sense often shares the dorsal column pathway, the second order neurons, and enters the medial lemniscus. But pain and temperature in particular travels within a separate ascending pathway called the trigeminothalamic tract. This will ascend to the levels of the thalamus, where it synapses with separate nuclei there, and eventually third order sensory neurons emerge from the thalamic nuclei and transmit the sensory signal on to the somatosensory cortex. Okay, so again, this is a pathway only found in the brainstem, all right, but it is the means by which general somatic sensation from the head and face area, the oral cavity, nasal cavity, is relayed to the thalamus, and then onward from the thalamus to the somatosensory cortex. So we've looked at the ascending or sensory pathways running through the main pathways running through the spinal cord. We're going to now briefly look at descending pathways and tracts. So these are carrying efferent or motor signals originating from the brain and delivered to spinal cord motor neurons. We can divide this group into two separate functional pathways, the direct pathways and the indirect pathways. Direct pathways are the most common of these is the pyramidal tracts, which is also known as the corticospinal tracts. Okay, but there's a subdivision of this grouping called the corticobulbar. Bulbar in this case means brainstem, and that's particularly applicable to how we activate, for example, the motor nuclei in cranial nerves. And there's a third tract that we'll talk about, the rubrospinal tract, which technically is an indirect pathway, but actually it works very closely with the corticospinal tracts. Direct pathways mean, and direct in this case means that it goes all the way, usually from the motor cortex in the brain, all the way down to end on in the ventral horn of the spinal cord, without making another synapse. The indirect pathways are pathways which there is at least one relay, perhaps more, going from motor cortex, which is the origin of the signal, before it reaches the level of the anterior or ventral motor horn in the ventral horn of the spinal cord, where the motor neurons are located. So the indirect pathways in this case, in the origin for nearly all of the descending fibers or descending axons that enter these tracts are within the brainstem motor nuclei. And so they would include pathways such as the rubrospinal tract, originating out of the red nucleus, the vestibulospinal tract, originating out of vestibular nuclei in the pons and the medulla of the brainstem, the medial and lateral reticulospinal tracts that we talked about again in connection with the pons and the medulla, or the various segments of the midbrain, these are the descending tracts that emerge out of those reticular nuclei within the brainstem. And the tectospinal tract, which comes from the posterior part of the midbrain and is involved with coordinating the head and neck movements and visual or movements of the eyes to follow a visual signal or pattern. There's two other terminologies we should talk about briefly in connection with these descending pathways and tracts. So these motor pathways involve two separate or main other groups of neurons, the upper motor neurons and lower motor neurons. The upper motor neurons are the brain neurons that are the origin points for these long descending motor fibers or motor axons. They are, in the case of the corticospinal tracts, these are the pyramidal cells which are located in the primary motor cortex, that posterior part of the frontal lobe. And these are the cells of origin for the pyramidal tract or the corticospinal tract. The brainstem motor nuclei, these are the points of origin for the indirect motor pathways, and these are both considered to be upper motor neurons. Okay, so these are the origin points for these descending pathways or descending tracts in the brain. The lower motor neurons these are the motor neurons which are located in the ventral horn, whose cell bodies are located in the ventral horn of the spinal cord. These are the somatic motor neurons that will innervate or supply skeletal muscles and activate skeletal muscles. Okay, so that's the difference between those two terms. So the direct pathway, or what we commonly refer to as the pyramidal pathway, which is also known as the corticospinal tract. Okay. These tracts, again, generally descend without synapsing. They originate from motor neurons in the primary motor cortex. They descend usually without synapsing all the way down to the ventral horn of the spinal cord, where they end on either interneurons or lower motor neurons or ventral horn motor neurons in the ventral horn of the spinal cord. This pathway involves regulating fast and precise or skilled movements, and these fibers are mainly distributed to the more distal limb muscles. Now, there's a special sort of subdivision of the pyramidal tract, which really doesn't fit the classic definition we've given up here, but the cortical bulbar tracts, they play the same role as the corticospinal tracts, but instead of running all the way down to the ventral horn of the spinal cord, they instead terminate on somatic motor nuclei of cranial nerves within the brainstem. And these somatic motor nuclei and the cranial nerves will then innervate voluntary skeletal muscles in the head, neck, and facial regions. So again, this would involve the motor neurons which control the facial muscles of facial expression, or the motor neurons which, for example, control the muscles of mastication, branches of the trigeminal nerve. And so this is just a diagram illustrating the direct motor pathway, in this case, or the pyramidal tracts, the pyramidal cells in the primary motor cortex, and they enter these descending pyramidal or corticospinal tracts, and they descend all the way through the brainstem, crossover at the level of the medulla in the case of the lateral corticospinal tract, and end on motor neurons in the ventral horn of the spinal cord. Activate those motor neurons, and then once those motor neurons are activated, they will subsequently signal the skeletal muscle and trigger its contraction. So the indirect motor pathways, these are multi-neuronal systems typically. There will be several different neurons involved in transmitting the motor signal, going from brain to eventually ending on ventral horn or lower motor neurons in the spinal cord. Most of these descending axons emerge from brainstem motor nuclei. These include all of the descending motor pathways except the corticospinal or pyramidal tracts. These brainstem motor nuclei that are the origin points for these tracks are stimulated or activated through the premotor or primary motor cortex, the cerebellum, or in some cases by basal ganglia nuclei. One of these tracks, the rubrospinal tracts, is by virtue of its anatomy, it's technically fits into this classification of the indirect motor pathway, but functionally, in terms of the muscles that it's distributed to and the types of motor tasks that it regulates, this tract is really an extension of the corticospinal or the direct pyramidal system. Okay. So the rubrospinal tract emerges from the red nucleus in the midbrain. It descends, and it can be stimulated by premotor or primary motor cortex fibers from the cerebellum or from the basal ganglia nuclei in the lower part of the cerebral hemisphere. They can all stimulate and activate this red nucleus fiber grouping. The fibers descend, enter the rubrospinal tract, which descends through the lateral funiculus and eventually ends on ventral horn motor neurons, and which activates skeletal muscles, particularly skeletal muscles controlling the distal limbs, and which are associated with precise or more precise and finer movement patterns. So it's in that sense that it's related more to the pyramidal tract system than it is with the other indirect pathways. The rest of these indirect pathways, which would involve the vestibulospinal tracts, the medial and lateral reticulospinal tract, tectospinal tract, these are more involved with controlling trunk and proximal limb muscles, and the degree of regulation is less precise and less fine. So again, these pathways tend to regulate the axial or trunk muscles that are involved with maintaining balance and posture, and again, in that sense, the reticulospinal tracts and vestibulospinal tracts are the main tracts related to that function. The muscles which involve coarser limb movements, the proximal limb movements, again, it's more the medial and lateral reticulospinal tracts, or they involve movement of the head, neck, and eye in order to follow objects in the visual field, and that would be the tectospinal tracts. Okay, so this completes our look at the central nervous system and both the ascending and descending tracts within the spinal cord. In a general sense, this is not an area we're going to expect you to know a whole lot of, but it is an area that you should at least be exposed to, so that you've got some basic understanding of how these various fiber tracts are organized, and in terms of the motor tracts, how they work, with the direct pyramidal system being involved more with controlling the finer, more precise movements, and the indirect fiber pathways being involved more with controlling trunk muscles and proximal limb muscles that require a lesser degree of precision in terms of their activation and regulation. All right, so next week we'll be moving on and looking at the peripheral nervous system. Enjoy the rest of the week.